Ramachandran plot. The Ramachandran plot, also known as Ramachandran diagram, is a graphical representation of the torsional angles phi and psi of amino acid residues in a protein. The plot was developed by Dr. G.N. Ramachandran in 1963 as a way to analyze the conformation of polypeptide genes. The Ramachandran plot is an essential tool in protein structure analysis and has become a standard in the field of structural biology. The Ramachandran plot consists of a scatter plot of phi and psi angles of all residues in a protein structure. The x-axis represents the phi angle and the y-axis represents the psi angle. X-axis represents the phi angle and the y-axis represents the psi angle. The scatter plot is divided into regions which correspond to the allowed. X-axis represent the phi angle and the y-axis represent the psi angle. The scatter plot is divided into regions which correspond to the allowed, disallowed and rare conformational states of amino acid residues. The allowed regions of the plot represent the conformational angles that are energetically favorable and compatible with stable protein structures. Disallowed regions of the plot correspond to phi-psi combinations that are energetically unfavorable and likely to result in structural instability. Rare regions of the plot are intermediate between allowed and disallowed regions, representing conformational states that are energetically possible but less frequently observed in protein structures. The Ramachandran plot provides valuable insights into the conformational states of protein structures. For example, the plot can be used to identify structural motifs such as alpha helices and beta sheets and to detect structural deviations from these motifs. In addition, the plot can be used to access the quality of protein structure generated by computational methods such as molecular dynamics, simulations, or homology modeling. Deviations from the allowed regions of the plot can indicate structural inaccuracies or errors in protein structures. The Ramachandran plot has become an integral part of protein structure analysis and is widely used in the field of structural biology. It has contributed to our understanding of protein folding and probability stability and has enabled the design of novel protein structures with desired properties. The Ramachandran plot continues to be a valuable tool for protein structure analysis, and its use is likely to expand as computational methods and experimental techniques continue to advance. The Ramachandran plot continues to be a valuable tool for protein structure analysis, and its use is likely to expand as computational methods and experimental techniques continue to advance. Advantages of Ramachandran plot. The Ramachandran plot has several advantages in the field of protein structure analysis. Advantages of Ramachandran plot. The Ramachandran plot has several advantages in the field of protein structure analysis. Provides a visual representation. The plot provides a visual representation of the conformational angles of amino acid residues in a protein. This makes it easy to identify structural motifs such as alpha helices and beta sheets and to detect deviations from these motifs. Allows assessment of protein structure quality. Allows assessment of protein structure quality. The plot can be used to assess the quality of protein structure generated by computational methods or experimental techniques. Deviations from the allowed regions of the plot can indicate structural inaccuracies or errors in protein structures. Enables identification of rare conformations. The plot can identify rare conformations of amino acid residues that are intermediate between allowed and disallowed regions. These rare conformations can be important for understanding protein folding and stability and for designing novel protein structures. Provides insight into protein folding. The plot has been used to study protein folding and stability and has contributed to our understanding of these processes. It has also enabled the design of novel protein structures with the desired properties. Next is the standardized tool. The Ramachandran plot has become a standardized tool in the field of structural biology as it widely used by researchers worldwide. This ensures consistency in the analysis of protein structures and enables comparisons between different protein structures. Overall, the Ramachandran plot is a powerful tool for analyzing protein structures and has contributed significantly to our understanding of protein folding and stability. Its widespread use and standardization make it an indispensable tool in the field of structural biology. What are the disadvantages of Ramachandran plot? While the Ramachandran plot is a valuable tool for analyzing protein structures, there are also some limitations and potential disadvantages to consider. Limited to backbone conformation. The plot only considers the torsional angles of the protein backbone and does not provide information about side chain conformation. Before moving on to the disadvantages, let's quickly summarize what are the advantages. Provide a visual representation, allow assessment of protein structure quality, enables identification of rare conformations, provides insight into protein folding, provides insights into protein folding, standardized tool, it's a standardized tool. Limited, what are the disadvantages? Limited to backbone conformation. The plot only considers the torsional angles of the protein backbone and does not provide information about side chain conformation. Therefore, it may not capture all aspects of protein structure. Not applicable to non-protein structures. The plot is specific to amino acid residues and cannot be applied to non-protein structures such as nucleic acids or carbohydrates. Limited to single residues. Limited to single residues. The plot represents the conformational angles of individual residues in a protein and does not provide information about interactions between adjacent residues or larger structural features. Limited accuracy for rare conformations. The allowed regions of the plot are based on statistical analysis of protein structures and may be accurately present rare conformations that are important for specific proteins or functions. Limited use in protein design. While the protein plot can help identify rare conformations and structural motifs, it may not provide enough information to design new protein structures with specific functions. Overall, the Ramachandran plot is a valuable tool for analyzing protein structures, but it has some limitations and should be used in conjunction with other analytical techniques to gain a comprehensive understanding of protein structure and function. Application of Ramachandran plot. The Ramachandran plot has several applications in the field of structural biology, including protein structure validation. The plot is used to validate the quality of protein structure generated by experimental or computational methods. Deviations from the allowed regions of the plot can indicate structural inaccuracies or errors. Identification of structural motifs. The plot is used to identify structural motifs such as alpha helixes and beta sheets and to detect deviations from these motifs. Prediction of protein stability. The plot can be used to predict the stability of protein structures by analyzing the distribution of torsional angles in the allowed regions. Protein design. The plot can be used to assign novel protein structures with specific functions by identifying rare conformations or structural motifs that are relevant to the desired function. Identification of protein folding intermediates. The plot can be used to identify protein folding intermediates by analyzing the distribution of torsional angles during the folding process. Analysis of protein-protein interactions. The plot can be used to analyze protein-protein interactions by identifying conformational changes in protein structures upon interaction. Analysis of protein dynamics. The plot can be used to analyze protein dynamics by tracking changes in torsional angles over time or in response to environmental changes. Overall, the Ramachandran plot is a versatile tool that has many applications in the field of structural biology. Its widespread use makes it an essential tool for analyzing protein structures and understanding protein folding, stability, and function. Let's have a quick summary. Developed by G. and Ramachandran in 1963. So, which are the major points of advantages of Ramachandran plot? Provide a visual representation, allows assessment of protein structure quality, enable identification of rare conformations, provide insights into protein folding, standardized tool will be there, provides insights into protein folding, standardized tool will be there. And what are the disadvantages? Limited to backbone conformation, not applicable to non-protein structures, limited to single residues, limited accuracy for rare conformations, limited use in protein design. These are the disadvantages. What are the applications? Protein structure validation, identification of structural motifs, prediction of protein stability, protein design, analysis of protein-protein interactions, analysis of protein dynamics. These are the applications. Thank you for listening.